Для следующего доклада на сцену приглашается лауреат Нобелевской премии Уильям Мёрнер. Уильям Мёрнер, профессор из университета Стэнфорда, Соединенных Штатов Америки. В 1978 году в Корнельском университете защитил диссертацию по лазерной физике. Член Национальной академии наук Соединенных Штатов Америки, Американского химического общества и множества других научных сообществ. Область его интересов включает исследование физической химии и биофизики отдельных молекул. Под его руководством активно развиваются методы двух- и трехмерной визуализации сверхвысокого разрешения для решения задач клеточной биологии, таких как визуализация и определение структур белков в бактериях, изучение организации хроматина, исследование динамики регуляторных белков в органах движения микроорганизмов, изучение молекулярного движения отдельных молекул белков, ДНК и РНК в реальном времени, высокоточный анализ фотодинамики отдельных биомолекул в растворе, применение к исследованиям процесса фотосинтеза, белковых взаимодействий и транспорта. Профессор Мёрнер удостоен множества престижных научных наград, включая премию Ленгмюра в химической физике и премию Дебая в физической химии, а также Нобелевской премии по химии 2014 года за разработку флуоресцентной микроскопии высокого разрешения. Пожалуйста, профессор Мёрнер.
Well, I'd like to begin by thanking the organizers and my hosts for inviting me to come and speak to you today. It's a great honor and a privilege to help celebrate the 150th anniversary of Mendeleev's great, wonderful contribution, the periodic table. Of course, I was promised a microphone that I can walk around with, and it's sitting right here. So maybe we should set it up. Huh? And then I'll be able to walk around a little bit. Okay, maybe I can get this on. Oy, oy. This is really difficult. It's another challenge. As we're beginning, test 1, 2, 3. Okay, it's the best we can do. We have this wonderful pointer, but unfortunately you cannot see the pointer. And I'm disappointed about that because I was not allowed to use my own computer, and if I were using my own computer, you would see the pointer because it's built into the display. So I'm sorry about that. In any case, I'm going to tell you about this story of single molecules. But first, let's talk a little bit about this great day that we're celebrating and Mendeleev. You see, what was so exciting about Mendeleev to me is that he had this deep desire to understand the world by working to uncover patterns. And this has inspired all of us ever since in many ways. Actually, for me, learning quantum mechanics, which I had to do as a physical chemist, because that's what I am, a physical chemist, and the fact that it would, in a fundamental way, explains the patterns in the periodic table, that's a truly beautiful thing. And that's what led me to chemical physics eventually and physical chemistry. So now, the roadmap for my remarks here are going to be this story of physical chemistry and ultra-sensitive detection. and how industrial basic research led to these advances, having unexpected effects spanning physics, chemistry, engineering, and biology. So the early days of single molecule detection and spectroscopy will be the beginning. Then I'll talk about something called super resolution microscopy with single molecules, and then try to explain what can we learn if you can look at individual molecules. So that's the roadmap, and so let's get started back in the very early days. So thinking about resolution is part of the problem here today, and so to get all of this on in your mind, resolution means the minimum distance in which you can distinguish two objects. And if you think of many, many orders of magnitude, you know that starting here at 100 microns on the far right, that's about the size of human hair, but going down by factors of 10, there are many, many interesting objects you'd like to understand. Cells, bacteria, and so forth, viruses, molecules, even single organic molecules, and atoms at 1 angstrom. So to observe these objects, you can use a lot of different techniques. You can use, on the left, these techniques, AFM, NMR, and electron microscopy, which have very high resolution, but they don't work in live cells, for example. And it's hard to see specific objects because you don't have a way to label individuals to distinguish them from the others. Light microscopy, on the other hand, is very non-invasive, and you can specifically label a biomolecule and then know which object you're observing. But the problem with using light is that the diffraction limit allows us to not see small structures beyond this boundary of about 200 to 300 nanometers, the wavelength divided by 2 times the numerical aperture of the microscope, which is about 1. That's the so-called diffraction limit. And so this whole idea of super resolution microscopy allows us to go beyond this barrier and obtain resolution down to 100 nanometers, 50 nanometers, 20 nanometers, and so on. So the breaking of the diffraction limit is the key thing that we'll learn about as my talk goes on. All right, so unfortunately, back in the 1980s, when we were thinking about detecting single molecules, Erwin Schrödinger, a great founder of quantum mechanics, said that we never experiment with just one electron or atom or molecule. And if we did, it would be as ridiculous as having a dinosaur in the zoo. So this is, of course, something that I didn't know about. It was published in 1952, but I didn't know about this when we did our first work, thank goodness. But the message for you today is, this is a few signposts that will appear up here. Beware when Nobel laureates say something cannot be done. Okay, so that just inspires people to go beyond. So in the mid-1980s, single ions were detected in vacuum and electromagnetic traps. Single atoms were studied with scanning tunneling microscopy on a surface. But what about molecules? People thought it was impossible, of course. So I want to tell you how we came to the point of detecting a single molecule. So we need a little spectroscopy now. Think of a molecule like pterylene shown here, highly conjugated aromatic hydrocarbon. And if you put the molecule in a transparent host of paraterphenyl, then measure its spectrum at room temperature, its optical absorption spectrum, or UV-Vis, you see the lowest electronic transition on the right, and then some vibronic progression. So broad at room temperature, as you know. But when you cool this to low temperatures, 2 degrees Kelvin, then that first electronic transition gets very narrow, so narrow that you can see 4 inequivalent sites in the host crystal. And so now let's think about just one of those very narrow lines. It turns out it's filled with many, many more tiny narrow lines coming from the individual molecules, but you can't see them here. So in those days, I was at IBM Research in California, San Jose, California, and we were using this kind of a spectral line at low temperatures, the kind that I just showed you there on the right. That kind of spectral line is going to be useful because it can allow something called frequency domain optical storage. The idea was to store bits of data in that line by marking different colors or different wavelengths using a laser and changing the absorption of the molecules, a method called spectral hole burning. And this actually utilized the fact that these were inhomogeneously broadened lines of zero phonon transitions. And much great early work, foundational work, was done by Roman Parsanov in Moscow and also Karl Rabane in Estonia. And so this work relied on this inhomogeneous broadening. But at that time, I was working at this great industrial research lab, and the nice part about it is that we not only were thinking about certain technologies, but we could also do the science underlying the technology. So in this case, the critical step was asking a question about these inhomogeneously broadened lines. So here's that inhomogeneously broadened line again, just like the one I showed you on the previous slide. Now we're spreading it out a little bit more. And the question is it smooth? Is it smooth like it's shown here? Now switching to penacene and paraterphenyl. No one had looked closely to see if it was truly smooth. So we looked to see, and we saw this. We saw this amazing structure, which we called statistical fine structure. And this statistical fine structure is coming from the fact that at each wavelength, there are different numbers of molecules piling up at that wavelength. And they're never going to be exactly uniform because of number fluctuation, square root of n effects. So, sorry about that. So this statistical fine structure showed us, we can see something that depends upon the number of molecules, actually the square root of the number of molecules. And that's my postdoc at the time, Tom Carter. So the idea here was, we've observed some strange new effect, and in fact, it's coming from the molecules and their individuality. Why don't we try to go see and detect a single molecule? So that's what happened in 1989, now 30 years ago. So this is another big anniversary for us. We detected the optical absorption of penacene in paraterphenyl at low temperatures. That's with my postdoc Lothar Kador. And it was done with quantum-limited laser frequency modulation spectroscopy, this fancy technique. But it worked to observe and open up the field. A year later, Michel Horry did a very important experiment. He also detected the absorption of a single molecule of penacene in paraterphenyl. But he used a slightly different detection. He detected the fluorescence emitted from the molecule as it was absorbing light. So that's an important step forward. So at this point, what have we got? We've got a low temperature, very high resolution, seemingly very esoteric thing, detecting a single molecule. A wonderful scientific advance, many scientific effects, but some people might have thought, well, who cares? Why did you want to detect these single molecules? So the first reason is surprises start to occur. On the left, you're going to see many spectral scans over a little spectral range, and then you will see, hopefully, if something goes wrong, where is it? It should be there. It's not starting. That's too bad. Let's try one more time. It worked briefly during the testing period. All right. Anyway, what we observed is that a few individual molecules, instead of sitting at one frequency, they would start jumping around from one frequency to another in a stochastic sort of way. And if you fix the laser at a fixed frequency, then you see that the molecule is in resonance, out of resonance, in resonance, out of resonance. It's like it's blinking on and off, turning on and off in a random sort of way. Again, people thought, well, this is not very interesting, but maybe we'll have some application later. The other thing that we were able to observe is that you could use light to move the molecules around. You can have a single molecule sitting there at one frequency and then irradiate it and it disappears and then wait a little while and it comes back again. So we were able to use light to control the molecules, another interesting effect. The message in this part of my talk is that even in an industrial research environment, when the exploration of technology includes a passion to explore fundamental basic science, important discoveries can occur. So now, the next question that you might be wondering about, well, why do this? Who cares? Why should you study a single molecule? So let me try to illustrate the answer, which is, we don't then have to measure only averages. Remember, all previous experiments before single molecules were averaging over millions and quadrillions and billions of molecules at the same time when you do a measurement. Now you can measure one at a time and measure it, and see if they're all the same or if they're different. The analogy here is a sort of a simple one. Think about this picture of Berlin from far away, and you're trying to observe cars, so you try to look closer. And now, yes, you know there's cars on those streets, but you're now measuring the average. You're not seeing any individual cars. You try to get closer. It's still pretty much averaged, and you cannot see what's really happening. But if you have a method of seeing the individual cars, then you see that some are stopped, some are moving, some are maybe turning, some are doing different things. And this is the kind of thing that can happen when you can observe individuals instead of just the averages. So that's why we do this. Now, how do we image a single molecule? So first of all, we're pumping electronic transitions, a ground to excited state, electronic transition. Now we're going to collect the emitted fluorescence terminating on vibrational levels of the ground state. And if we do that, we're going to be able to see each single molecule as a dot. And so think of it this way. A laser is sent through a microscope. It's focused with an objective, which I cannot point to. Then the emitted light is shifted to long wavelengths due to the Stokes shift. And so the detection system, which is a camera or so forth, needs filters to block out the pumping laser light in order to detect the light from the molecule. And it's worth noting right here that even your own eye, you can look into my microscope and you can see the light from these single molecules. Your eyes are sensitive enough to be able to detect the light from individual molecules. Of course, we have to have the right laser color, and we have to collect well, and we have to do various things to make this work easily, but it's done now all over the world in many laboratories, including, of course, in Russia. So, thinking about this from a slightly different way, let's look here at the picture on the right, and you see most often, maybe you want to study a protein that's not fluorescent. Then what we will do is you'll attach a small fluorescent dye to that molecule, and then it's going to be the light from that fluorescent label that you're going to be detecting. So these labels are rhodamine dyes, cyanine dyes, other good emissive dyes, fluorescent proteins like green fluorescent protein, even antenna proteins from photosynthesis. But now imagine you're focusing your laser to a small spot, and remember that diffraction effect that I mentioned a few moments ago. It limits the minimum size of that spot to a few hundred nanometers. So the molecules are 1 nanometer in size, but this laser spot is a few hundred nanometers in size. So now you see that to pump a single molecule, you have to dilute them. You have to make sure that they're farther apart than the size of this laser beam. So if you do that, you'll be able to detect single molecules done carefully. And here's an example, hoping that this important video, yes. So this is now in a cell, and I'm looking at a transmembrane protein that's important in your immune system. It's been labeled, and you can see at room temperature, there's a spectacular dance of these molecules on the surface of the cell. Much information is hidden in that motion. How important is cholesterol? What is the diffusion coefficient? Is it normal motion or not? And let's look at it again, because this little movie, which is some kind of old, it's still really beautiful to me, because not only do you see the motion, you also see some of the molecules disappear. What does that mean? You notice in one step they disappear. That's a proof that it's a single molecule, but this is photobleaching, okay, on the single molecule level. You don't see an exponential decay. You see the molecule simply disappear, and that's some proof that it is an individual molecule. So there's other things in that movie, but because of time, I'll just keep going. So now that people started studying single molecules at room temperature in the mid-1990s, surprises started to appear. Again, just like at low temperatures. So what surprises? On the left are bacterial cells in white light. So I'm just showing you in white light a few bacterial cells. They're pretty small. But on the right, I'm showing you the light coming from that sample. And in these bacteria, a particular protein has been fused to yellow fluorescent protein. So this fluorescent protein is a label, and you're looking at the light from the label, right? What happens, though, if I just run the movie and show you what happens? It's amazing. These molecules are blinking. This was the surprise. We didn't expect this for fluorescent proteins at room temperature. So each one of these flashes of light are coming from inside of those bacterial cells. And the movie contains a great deal of information about all of those individual single molecules. And here there's no motion. So please don't be confused. That earlier movie was showing motion moving around. Here there's no motion. All of this is just the blinking of fixed molecules, which are going to be useful in a few minutes. But you see, again, people said, oh, what good are single molecules? They blink too much and so on. It's not so interesting. Well, we'll see. But when you explore a new regime, it's a wonderful aspect that you really can't predict in advance some of the surprises that might occur if you hadn't done the experiments. This is why basic science is so essential. Doing the experiments, you can find out if there are surprises. And the surprises can lead to some unexpected and unplanned applications. So I'll tell you about that. Now let's start talking about the bigger problem, okay? The big problem here is we'd like to observe in these cells, in this case a bacterium, for example, all of these nanoscale machines that are at work inside. I mean, you can see DNA and proteins and so on. Looking more closely, you can see motors, like the motors that were mentioned in the Professor Savage's talk. And you'd like to be able to see what they're doing to learn about this interesting system. So you want to have a technique that has high sensitivity and high specificity, and possibly living cells, and high spatial resolution. Because look at the scale bar here. You see that little 20 nanometer. These objects are really tiny. So you might say, well, you could use electron microscopy, but in electron microscopy, all of this just becomes a fuzzy blur of gray objects, gray and white objects. So you want some specificity, and that's what fluorescence does for you. It lets you label a specific set of molecules so that the light only comes from them. However, here's the problem. The smallest possible visible laser spot is too big compared to the size of all these objects. The diffraction limit, again, is limiting it. So that's what led to super-resolution microscopy, and Stefan Hell used a particular technique called stimulated emission depletion. Eric Betzig and I used single molecules in the way I'm going to describe now. So once again, explaining what we're talking about. Here's the bacterial cell again, and we've labeled now a particular protein with these fluorescent proteins, and you're seeing the light, and you want to see the structure. You might think all you have to do is to buy the most expensive microscope possible. So here it is. Here's this most expensive microscope, and you don't see anything. That's because each of the individuals, even though they're only a few nanometers in size, they appear to be a few hundred nanometers in size due to that Abbe's diffraction limit. So when you use super resolution, however, this image becomes that image. And that's the difference. Before, you could not possibly see the detail, and there was no way to make it better because it was fundamentally blurry due to the diffraction limit. But with super resolution, we can remove that limitation and see this detail down to the 40 nanometer scale in this case. So how does it work? The first idea is a simple one: super localized single molecules. Think of this little mountain, which is in a lake in Oregon. And you know that if you just walk up to the top of the lake with your cell phone, you can just simply read out the coordinates of the top of the mountain. And of course, I have to have a scale bar there. It's 120 times 10 to the 9th nanometers, okay? So let's see if anybody's awake. In any case, the location is what you want to measure. So our individual molecules, in our microscope now, the bar is 250 nanometers. It looks like a little mountain if I plot the signal in the z direction, and I want to find the position of that molecule. So what we do is to fit the shape. We've measured the shape in the microscope, and we use a function, a Gaussian function, and others to determine the position of the molecule. Then so for example, in 2 dimensions, I'm showing the image of the molecule. With some fitting of data, we find out where it is. So that's the first key idea. And this works to a precision that is the Abbe limit divided by the square root of the number of photons detected. If I detect 10 to the 4th photons, then I can localize to 100 times below the diffraction limit. Instead of 200 nanometers, in principle, it would be 2 nanometers in terms of the position. But this only works for one molecule when it's separated from all the others. And this is not resolution, it's only localization. So to get resolution, you have to do one more thing, and I like to call it active control of how many molecules are on at any given time. So the idea is we have a structure we want to see, and you put fluorescent labels all over it. If you allow them to all emit at the same time, then you get this very blurry, fuzzy picture. And so that's the idea. You don't let them all emit at the same time. You actively make sure that only a few are on. Using an on-off process, using either photochemistry or blinking, like I was just describing a moment ago. I start out dark and then only allow a few to emit. Then I can localize those with the upper trick and then photo bleach them, and then you localize another group randomly selected, photo bleach them, and you keep writing down these positions of all these molecules. In the end, you take all the positions and show them at the same time, and you see structure beyond the diffraction limit. So that's how it works. And this has been called many names with many acronyms. I won't go through all of these acronyms, but a mechanism-independent acronym that we like a little bit is called Single Molecule Active Control Microscopy, or SMACM. All right, so that's how it works, but let's try one more time to make sure everybody understands how this works. If you will, maybe you can explain this to your children or your family. Think about fireflies for a minute. Fireflies. You've all heard of fireflies, right? People have heard of fireflies. Yes? Yes? Хорошо. So, now imagine you have a tree. You have a big tree, and the tree, but it's nighttime, and you want to see the branches of the tree. So it's dark, though. You can't see them. So the idea is to place fireflies all along the branches of the tree. Okay? Place fireflies. And then just take your camera and take a movie of what's happening, because the fireflies will blink on and off, randomly at different positions. Every time you see one of them on, you measure where is it, and write that down, measure where is it, write that down, and then show them all the positions at the same time, and you'll see the branches of the tree. And that's how this works. And it uses those single molecules. So I'll show this to you a little bit differently here with molecules, the same idea. On the left is the diffraction limited image, one frame of the movie. This is now a mammalian cell, and we've labeled microtubules, and you see these dots, which are the single molecules from one frame of a blinking movie. And on the right, you see little tiny dots, which are the locations of all those molecules. Next frame of the movie, remember it's blinking. Next frame of the movie, blinking. And here, if you just run the movie, try again. There. You see that from all of that blinking, you get the information about the positions of the molecules and this image with very high resolution. So that's the idea. Now, as chemists, you want to think more about this process that lets us control the molecules, having them be on and off. And so I don't look at the long list of mechanisms that have been demonstrated. Just look at these energy levels for a moment. Remember, we want to create a fluorescent object that you can pump and emit in the middle. That's what you want to create, but in low concentration. So one way to do that is to start with a dark precursor molecule, a molecule that's not emissive, but then maybe a little control light will convert a precursor into an emissive molecule. And if you do that at low enough optical intensity, you'll create just a few of those molecules at any given time. That's one way to do it. Another way to do it is this blinking business, and blinking comes from an emissive process that's coupled to dark states on the far right. And so those dark states on the far right act as reservoirs to hold the molecules in the dark so they do not emit. And if you put most of them into these dark states, then every once in a while one will return and you'll see it emitting again. So this is what blinking is coming from a variety of processes, for example, photoreduction of the molecules with certain additives. And there's other mechanisms. Even to give you a little more flavor for the diversity in this area, but the need for more ideas. On the top, I'm showing you a precursor molecule that's converted to a fluorophore. That's because if you see the aromatic azide on the bottom left, that's not a very good donor in this donor-acceptor molecule. So a little irradiation in the blue will convert that azide. It will be basically blow off dinitrogen. There will be reactive nitrine left behind, and you get an amine. Not exactly the structure shown, but you can produce an amine from an azide this way, and now you turn on fluorescence. So that's been demonstrated. Another scheme is to label a molecule that you would call a ligand that binds somewhere that's important, like this molecule, saxitoxin, which binds to voltage-gated sodium channels on the surface of cells. Now you allow the molecules that are moving around in solution, you don't see them very well because they're fuzzy, because they're in motion. But then when they bind and sit on a channel, they sit at one place and you can localize the molecule that way. It's a technique called paint. Another example, here's a rhodamine spiralactam that can be opened by irradiation, which converts a dark molecule into an emissive molecule, and so on. But I want to say that there is a continuing need from our synthetic friends to come up with more molecules that we can control in terms of whether they're emissive or not. Okay, so now the next signpost. This super-resolution fluorescence microscopy is fascinating because it gives us a new window into the nanoscale. Because we're looking on the scale of these single molecules that are about a nanometer in size, and we can extract information based on how many photons we can collect from each molecule. The idea required fundamental scientific advances, uncovering this issue of can you detect a single molecule at all? And then what can you do with it? But also it required these surprises, the blinking and switching that I talked about, which lets us control the molecules. Examples now. Let's just enjoy a few cases. These are bacterial cells. 3 different proteins inside these bacteria have been labeled by fusing enhanced yellow fluorescent protein, EYFP, to the proteins of interest. Here's the diffraction-limited images. Okay? The diffraction-limited images don't tell you very much, but the super-resolution images are spectacular. The difference is just wonderful. With an object as small as a bacterium, it's already close to the diffraction limit in size. So you can see a lot when you just go below the diffraction limit by a factor of 5. And so that's one example. Another class of examples, and there's just too many for me to review because there's hundreds now. This is a case of mammalian cells in part of these pictures. On the upper left is a spectacular demonstration of something that was discovered by this super-resolution microscopy, which is the banding, the periodic banding of the spectrin molecules in an axon from the Zhuang lag. On the lower left are the very small aggregates from Huntington's disease. The amyloid diseases all have very small fibrils and aggregates that form inside cells. And many of the sizes are below the diffraction limit. This is actually observed with STED microscopy because we have also built a STED microscope. The upper right is bacteria again, but we're looking at something called the S layer that forms on bacteria and doing pulse chase, where the old S layer is in green and the new is in red. And with super resolution, you can see a great deal of detail about how this layer is formed. And the lower right, glycocalyx. The glycocalyx, something that many people haven't heard about. But all of our cells in our body almost are covered with sugars, covered with different glycans in different patterns. And these glycans are important for recognition between cells and interacting. They're elaborated in cancer and so on. And this is a super-resolution microscopic image of the surface glycans that have been detected by using methods from Carolyn Bertossi to attach fluorophores on specific glycans. You can see that they're hollow tubules. They're extending out from the membrane are these hollow tubules that form. And you can observe the shapes of these structures with this super-resolution microscopy. So I want to now go on to the next signpost. This super-resolution imaging is really an amazing new window into nanoscale structures in cells, 5 times, 10 times beyond the diffraction limit, and various people are coming up with even more ideas to push the resolution even further. But what does this do for us? It allows us to have a deeper understanding of cell biology, but also catalysis. People are using some of this for catalysis studies. Materials, polymers, shapes, and polymer mixtures or block copolymers. All of these interesting systems that have detailed structures below the diffraction limit can be observed using super resolution if you can label what you want to see. And these methods may lead to some important applications. For example, it's not only worth studying the normal cell biology, you can also study pathogenic cell biology. What happens with amyloids, what happens in cancer, and so on at this very, very tiny scale. So now, I want to talk a little bit more about one special aspect of these experiments, which is 3 dimensions. We'd like to be able to do all of these imaging tricks, including the Z dimension, not just X and Y, because you know the answer, the world is 3-dimensional, right? It's not 2-dimensional. If I'm trying to study some property like the motion of this transmembrane protein on the surface of a bacterium, but I can only observe it in 2 dimensions, you see that I only observe part of the motion, and miss a lot of the motion. So we've been working on techniques for 3-dimensional imaging, and here's one of them. So this thing on the left actually gives you, it's going to be a mind bender a little bit for some people. On the left is a standard microscope where all I'm doing is I have a single emitter and I'm moving it to different Z positions, and you see it's in focus at one time, but then it goes out of focus and it disappears very quickly. That doesn't help you very much. We change the microscope by just putting some simple optical components into the microscope. A little glass disc that has some different thicknesses in it. And by some beautiful magic, okay, of optics and all of these things that you've heard about, it actually comes from differential equations and the waves of light and so on. But anyway, a single emitter becomes 2 spots. 2 spots. And now if I move it up and down in Z, those spots revolve around one another. And that angle between those 2 spots gives me Z information. So this trick takes a conventional microscope and gives you X, Y, and Z. So you can use that to get 3-dimensional images like these, where the surface of bacteria are colored one color, and there's a protein inside that's colored with a different color. And this is really just playing with the light from the molecule. Since we understand how that light works, understand how it behaves as it propagates through space and so on, you can make these sorts of changes, and you can even make a single emitter look like a happy face, believe it or not. So here's one thing my students did. You've got some single emitters, and now we turn on this special optical component, and you see that all of a sudden no, please don't tell me. Why is my video not playing? Never allow a place to demand that you not use your computer. So these single molecules turn into a happy face, and then as you move above and below the focal plane, they turn into an unhappy face. Like my face right now. So using these 3-dimensional techniques, you can now look at structures inside bacteria on the left and see the full 3-dimensional shape of a helix-like structure. You can see structures at the ends of cells that certain proteins make. And on the far right, I'm hoping this one works. Does it work? The nuclear lamina? Yes, that one works. This is the nuclear lamina in a mammalian cell. So in almost all of our cells, there's of course a nucleus and then nuclear membrane. Right inside the nuclear membrane is the lamina. This is a 3-dimensional super-resolution image of the lamina, where you can even see a channel from one side to the other of the lamina itself at super resolution. So now, the last thing that I want to point out and just remind everyone so no one's confused. I've been talking partly about imaging of single molecules to produce a 3-dimensional super resolution image. That's an extended structure, and it needs to not be changing while we're measuring it. We want to measure that extended structure, and I've shown you many examples on the left. In turning that diffraction limited into this rotating image, which is not rotating. There it is. Okay, but let me just emphasize that this trick of 3-dimensional information also works for single molecules, of course, at low concentrations. So here I'm showing you what happens on the right with this thing that we call the double helix point spread function. And I'm showing you the motion of an object in 3 dimensions, even though the movie's not playing. That is a single RNA particle, actually, an RNA particle inside a cell nucleus that we can observe moving over a long time scale. And that kind of motion, of course, tells you about what's going on in the cell. Let me emphasize that a little bit more. If I can watch the single molecule move, then I will learn the science by just simply following its motion, measuring its motion, and understanding its motion. So here's one of many examples of this idea. A little bit more biology. In all of our cells, on the left, we have a structure called the primary cilium. The primary cilium sticks out from the cell, and it's a signaling center. It contains proteins that are involved in something like differentiation and other signaling between cells. So I'm showing you how a protein can come and bind and change the behavior of the cilium and actually change the genetic program of the cell. So this is a crucial process in differentiation, and also a crucial process in cancer, apparently, because these particular proteins become mutated quite a lot. So on the right, I'm showing you that we can image a single molecule. In this case, we're talking about this one that's green on the left. It's called patched, and a single patched in the cilium can be observed moving around and superlocalizing its position. And at the bottom, I'm showing you its position as a function of time, where the motion is part of the time gray, which is diffusion, part of the time is directed motion in blue, part of the time is binding in red, because it's moving and finding partners and doing things. It's being pushed by motors sometimes, other times it's moving by diffusion. All of that is hidden in the motion of one molecule. And we have a great challenge now to get as much information as possible from these kind of data, which involve mathematical analysis, statistical analysis, using methods that can extract that motion dependence in as much detail as possible. So I'm very excited to have had a chance to explain to you this field of single molecule spectroscopy and imaging and super resolution. The field has had impact in chemistry, in all the ways I list here and others, in physics and in biology. And working in this field has been extremely exciting for me over the past 30 years, because there are scientists everywhere in this global scientific enterprise that we're involved in who have made important advances to the field. I want to thank my past students, postdocs, and collaborators. And here's the current team. Here's our money and so on. At the lower left, you see a little logo, no ensemble averaging, if you like that. And I say that we're the guacamole team, right? The guacamole team. Well, what's a guacamole? It's one molecule, obviously. One molecule. 1 over avocado's number of moles. So with that very, very serious and sad joke, I want to thank you again for your attention and celebration of the Mendeleev 150th year's anniversary.